Effect of Stannius ligatures on frog's heart. In this experiment, we will learn about the normal pacemaker of the heart as well as the potential pacemakers in the heart. Principle: the rate of discharge is different in different potential pacemakers of the heart. We first pass a threaded aneurysm needle between the truncus arteriosus and the atria. We record the normal cardiogram on a slow-moving drum. Then we bring the thread forward under the truncus arteriosus and tie it at the junction of the sinus venosus and the atria. This is the first Stannius ligature. It separates the sinus venosus from the atria. We immediately record the cardiogram to study the effect of the first Stannius ligature on the cardiogram. Then we tie another thread between the atria and the ventricles. This is the second Stannius ligature. It separates the atria from the ventricles. We immediately record the cardiogram to observe the effect of the second Stannius ligature on the cardiogram. This is the normal cardiogram. At point A, we apply the first Stannius ligature between the sinus venosus and the atria. Because of this ligature, the atria and ventricles are cut off from the impulses from the pacemaker in the sinus venosus. Thus, there is a pause in the rhythm of the heart. But immediately, impulses are generated by the heart tissues in the atria. They generate a slower rhythm. The distance between two consecutive waves is more in the cardiogram after the first ligature. After tying the second ligature, the ventricles are cut off from the rhythm generated by the atria. Now the ventricles have to generate the rhythm for contraction. The frequency of contraction decreases after applying the second ligature. The ventricles take time to start generating the rhythm. That is why there is a long pause just after the second ligature is tied. This tells us that even though the sinus venosus is the normal pacemaker of the heart, tissues in other parts of the heart are also capable of generating impulses.